Good morning, everyone. Welcome to Grand Rounds. It's my pleasure today to introduce Dr. Paul Nghiem. After completing his undergraduate degree at Harvard University, Dr. Nghiem attended Stanford University for his medical degree, as well as a PhD in cancer biology. He returned to the East Coast for postgraduate training and completed a medicine internship at Brigham and Women's Hospital, and residency in dermatology at Massachusetts General, and a Howard Hughes Physician Fellowship in the Department of Chemistry and Chemical Biology, also at Harvard University. So Dr. Nghiem remained at Harvard University in the Dana-Farber Cancer Institute before coming to the University of Washington and the Seattle Cancer Care Alliance, where he's division head in the George Odland Endowed Chair in Dermatology, professor in dermatology in the Department of Medicine, and adjunct professor also in the Departments of Oral Health Sciences and Pathology. Dr. Nghem's focus of research is on basic skin cancer biology. He studies the checkpoints in the cell cycle that relate to damage from ultraviolet radiation, as well as the basic clinical and translational aspects of Merkel cell carcinoma, an uncommon but often lethal skin cancer that's associated with immune suppression, UV exposure, and viral infection. He's received a number of awards, the Young Investigator Award from the American Academy of Dermatology, an NIH Clinician Scientist Award, a Career Development Award in Skin Cancer from Harvard University, and as well as received the American Cancer Society Legacy and Leadership Award, which is entitled Finding Cures. Among his many involvements in professional organizations, Dr. Nghiem has served on the editorial board of the Journal of the American Academy of Dermatology, and has been elected member of the American Society of Clinical Investigation that recognizes physicians whose research has had an impact on the understanding and the treatment of major diseases. So please join me in welcoming Dr. Nghiem to speak on Merkel cell carcinoma, progress in the management of an aggressive immunogenic cancer.
Good morning. Thank you, Lauren. Very nice introduction. It's really a pleasure to be here. The Department of Medicine is a really special place that I'm very happy to call home. The theme of today will be that you can have fun, you can work as a team, you can bring people together and try and hope to find a problem where you can really make a difference. And boy, is that a lot of fun. And you'll see there's dozens of folks in this deep talent bench of the Department of Medicine that have been involved at the training level and at the faculty level with these studies. It's also a fascinating link, I think, between the immune system, a virus, ultraviolet light, and cancer, and the way that I think we're going to be treating a larger number of cancers and even infections into the future. Disclosures here, and the relevant, this is kind of a cute one. Anything I talk about in terms of therapy is off-label. Nothing has ever been approved for Merkel cell carcinoma by the FDA. But we hope that 2017 will change that, I think twofold over. An overview, here's an example of one of the relatively characteristic presentations of Merkel cell carcinoma, but I want to talk about the risk factors, ultraviolet light, and a very fascinating interaction with the immune system and with a virus that's extremely common on most of our skin, normal skin, on any given day. And then how we've used the role of the immune system and the virus that's usually incorporated into this cancer to both track and treat the cancer more effectively. And then therapy, really revolutionary stuff where I used to not basically need to see patients with metastatic Merkel cell carcinoma because they would get garden variety small cell chemotherapy and there'd be a short little bit of remission and then that was it. And it was a grim, grim time, and things are so different now. It's very exciting. A lot of hope. So why is this cancer important? It's 2 to 3 times more likely to kill somebody than, quote, the most lethal skin cancer, melanoma. That's because it's relatively rare, so people don't even list it as a skin cancer. But it's about 2,000 cases per year in the US, and that's not actually so rare. That's the same incidence of chronic myelogenous leukemia. It's quadrupled since 1986 in incidence, at least its reported incidence, and I'll tell you about that a little bit in a moment. The optimal therapy for the cancer is unique among all skin cancers in that its proper treatment is relatively benign and effective. but it's often not treated properly. So we'll get into that just a little bit. And then a new polyomavirus is associated with Merkel cell carcinoma. This was discovered in 2008, and that's just a fascinating cat and mouse game with the immune system and with ultraviolet as well. But first, I'll just tell you, how did this cancer get its name? This is a picture of Friedrich Merkel, a German anatomist, who in 1875 drew this picture of the dermal-epidermal junction of a rat paw. So this is the dermis down here, and this is the epidermis. And he saw these weird cells at the junction that were linked to nerves. And here's a more modern depiction of this. Here's the epidermal-dermal junction, and these cells, now called Merkel cells, were connected to nerves. And 135 years ago, he said, I think those might be touch cells. And he was exactly right. It took a long time to prove that, but they bear his name. And when cells that look like that turn into a cancer, they are unfortunately alarmingly banal looking on the outside. So let me tell you, don't worry too much about missing a Merkel cell carcinoma. Everybody will do it. It's 2/3 of them are missed. The doc, the day they do their biopsy, thought they were biopsying something benign, like a cyst. Like this absolutely looks like a cyst. So not to worry too extensively. The good news is, as soon as that biopsy is made, it's a very straightforward diagnosis under the microscope. All of these little blue guys are individual cells, and the brown perinuclear dots are cytokeratin 20. This was developed in about 1990s, and that totally changed. It made it much more straightforward to make this diagnosis. And it's very straightforward, and the good news is we can really do a lot after the diagnosis. I don't think it makes sense to try to educate people about this rare cancer that looks like a cyst beforehand. And the good news is there's a lot that can be done after the diagnosis. So here are many missed cases in the early, prior to the definition of the cytokeratin 20 antibody. But after that, these increases in incidence, now approaching right about 2,000 per year, are real. And they're really based on increases in risk factors, like all of the skin cancers. Prolonged sun exposure. This is overrepresented in people who don't make a lot of pigment, Caucasians, and are not thus protected by their melanin. It's very latitude associated. You get closer to the equator, you get a much higher incidence, and it tends to happen on sun-exposed skin. I'll show you a picture of that in a moment. But the early observation that this was epidemiologically linked to immune function was very intriguing. It's very strongly linked. Whether you take out T-cell function with a virus, HIV, with calcineurin inhibitors by a solid organ transplant recipient status, or you take out T-cell function by having a B-cell malignancy, chronic CLL, that's very associated with dysfunction of T-cells, you have a between 5 and 50-fold increase of getting this cancer and a couple-fold increase in dying of it. So very aggressive. In the immune-suppressed setting, although only 5% of patients with this cancer are significantly immune-suppressed in any way that we know of, besides being over the age of 50. And I want to tell you that there is a little bit of a link to immune suppression and being over the age of 50. And here's the relationship of age and Merkel cell carcinoma shown in red versus all cancer cases in blue. And of course, the denominator is very different, 1 million versus 2,000. But this shows the shape of the relationships there. And you can see that Merkel cell carcinoma that all cancers start to take off in the 30s, 40s. They actually fall off later in life, 80s and such. Whereas Merkel is fascinating delay. As I'll tell you in a minute, the virus, we're exposed to it in daycare. It's on the doorknobs. It's been there, all along. And there's a 5-decade delay between getting the virus and getting the cancer in the earliest settings. And then there's this amazing cooperativity with age. As you get older, it's really steep. And I believe this is the exact same phenomenon, immune senescence, that underlies why we get shingles from the varicella zoster virus after 50, 60, that kind of thing. Our T cells are a little bit less diverse and a little bit less effective, and I think that this combines with genetic mutations and gives us a really, really strong cooperative increase in incidence there. So here's a picture then of where it occurs on the body. These are all the black ones are primary Merkel cell carcinomas, obviously a lot more on the sun-exposed face than on the sun-protected scalp, the sun-exposed arms than the sun-protected parts of the arms. But just like melanoma, about 15% can happen where there's minimal or no sun exposure, buttocks, genitals. But then this I have a cute little thought experiment here for you. All these open circles are Merkel cell carcinomas arising in lymph nodes without an associated primary tumor. And my question here is, assuming that there's already palpable nodal disease, which is how all of these presented, is it better to still have that primary lesion or to not have a primary lesion? I tell you, the patients come in and they're distraught. They don't see a primary lesion. Their docs can't find one. And should you be reassuring them or telling them, yeah, you're in hotter water? And I'll come back to that concept in a minute. So how does one stage a patient with Merkel cell carcinoma? This is obviously a huge difference depending on stage, between a 90% survival and a 10% survival. And as of 2010, the staging by the AJCC was a complete mess. And there were 5 staging systems out there. None were really consensus. And so Bianca Lemos, who was an intern in this medicine program a few years ago, published a paper saying pathologic nodal evaluation improves prognostic accuracy in Merkel cell carcinoma, and that was the basis of the first international consensus staging system, looking at 6,000 cases or so. What does that mean? That means if you look in the lymph nodes pathologically, you get a much better sense of what the risk is for the patient. So you get 100 patients with, say, a Merkel on their arm. You feel their armpit. You can't feel anything. A scan shows it's negative. You are wrong 1/3 of the time. That rate is 30 times higher than for melanoma. You take your average melanoma and you feel the lymph nodes are negative and you got a 1% chance that you missed it. With Merkel, it's a 30% chance. And so that means if you don't look pathologically, and 68% of patients were not staged with anybody taking out their lymph node and looking, you're wrong a lot of the time, and that translates to this survival difference. 1A is pathologically negative nodes for a small tumor, and 1B is the doc and the scanner said they didn't see anything. And this is about a 4-fold difference in death. So that's quite significant. In terms of that's local disease. Then here's nodal and distant disease. And this new system broke out microscopic nodal disease with clinically evident nodal disease, which makes some difference. But now I'm back to my question. These are folks who have these are folks who have nodal disease that was grossly palpable at presentation and either a known or an unknown primary. And there is a 2-fold difference in survival as to whether you have a known or unknown primary. So you've thought about it a little bit, and I'll just tell you the answer now. You're much better off, and this is the exact same direction that happens with melanoma. You're much better off if you have an unknown primary. Here, the risk reduction is 70% or 60%, depending on univariate or multivariate analyses. It's a huge risk reduction to not have that primary anymore, and we strongly believe that the primary was eliminated by the immune system, and I'll show you why. So first of all, this is true across multiple large cohorts. Here, this is a Mayo Clinic group. This is our Seattle cohort of patients presenting with so-called 3B disease, nodal grossly evident disease. And here's the New York cohort. And you can see the purple line's twice as high as the gold line every time. There is almost twice the survival if you got rid of your primary lesion. And what do we think is happening? Well, if this is the positive node and here's the primary tumor that's still there, we believe in these cases the immune system was not able to get rid of that primary lesion, given that you're already in a bit of hot water and having a lymph node be clinically evident. In contrast, if you had that lymph node but your immune system managed to get rid of the primary, that's probably because they had highly functioning immune cells and maybe could control distant disease elsewhere. And one of the many reasons that we believe this is the case is when we look at our 3B cases and we plot them as a function of whether the patient was immune suppressed or not immune suppressed. You'd guess that the immune suppressed people, if the immune system was mediating this, would not be able to generate unknown primary lesions. And that's exactly what we saw. No one in our cohort who was immune suppressed managed to get rid of their primary lesion. Whereas the not immune suppressed folks did in the vast majority of cases. So I'm not really going to talk about early management of Merkel cell carcinoma because surgery and radiation have been getting refined for decades, and they're very nicely summarized in the National Comprehensive Cancer Network guidelines for Merkel cell carcinoma. And the bottom line is, probably 98% of the time, somewhere over 95% of the time, surgery and radiation get rid of all the cancer we can see. Patient's very happy. Nothing on scan. But it's going to come back in almost half. Very likely to have spread in an occult way. And what did we always do for that before? It was chemotherapy with a small cell regimen. This is absolutely the standard around the world until very recently. It shrinks the cancer in about 2/3 of cases. And this is the problem for pharmaceutical companies. They're like, how do we go up against something that works 2/3 of the time? That was a problem. They were kind of scared. And it's a rare cancer. But then by 90 days after starting chemotherapy, most patients have had their cancer come back, their immune system is suppressed, their cancer is broadly resistant to chemotherapeutic agents, and life is short at that point. It is not good at all. So we really needed better therapies and a better understanding. So given these links to the immune suppression, to the ultraviolet, how does Merkel cell carcinoma arise? And I already kind of told you the punchline here. In 2008, a new human virus that causes cancer was discovered by Patrick Moore and Yuan Chang, a dynamic duo, husband and wife team at Pittsburgh now, who, when they were in New York, also discovered the Kaposi sarcoma herpes virus in the 1990s. So between the two of them, they have found 2 of the 7 viruses that cause cancer in humans. And this has been broadly validated across thousands of patients and dozens of studies. And it's really clear that this virus is necessary for causing this cancer in about 80% of cases. And as I told you, this virus is, if we swab your skin this morning, most people will have PCR detectable Merkel cell polyomavirus on their normal skin. What? We have a virus all over the place, in the daycare centers, on the doorknobs, and only 1 in 3,000 people over their lifetime will get this cancer? How is that possible? It's a perfect storm. Very simple, as it were. We understand now. There's a virus. There's the immune system. There's infection as a child. The virus hangs out in the cytoplasm, does not go into the nucleus, does not integrate into the genome, and you get antibodies to the outer coat proteins, which I'll be talking about, the capsid proteins, as evidence of specific infection early. No problem. No lesions on the skin. No one's ever found a primary lesion of this infection. If 2 rare events happen that neither the cell nor the virus want to have happen, then that's the necessary next step for getting this cancer. Number 1, a clonal integration event somewhere in the genome, different in every patient, that the virus gets in there, and what it would normally do then is make hundreds of copies of itself at that integration site, and the cell would say, oh, I've got 200 copies of chromosome 2, I'm going to kill myself. Genomic fidelity machinery turns on, and therefore a second machinery has to be activated right away to chop in half, perhaps by ultraviolet mutation, perhaps by luck, whatever, to chop in half the critical oncoprotein that drives the cancer. This is the oncoprotein that takes out tumor suppressors and pushes the cell into the cell cycle, and then it kills the other half of that protein that would cause replication initiation at that particular site. So that's why this is really rare. Both of those events have to happen. They're different in every cancer, and otherwise we would have multiple Merkel cells all over our body. And then this rare thing happens, and then here's the expression of the viral antigens, and then the final piece of the puzzle is the immune system has to be duped. And that's what we've been spending a lot of time understanding and overcoming. But I want to tell you now what we've learned by doing genetic analyses on the virus-positive Merkels. And you remember I told you there's about 20% virus negatives. Interestingly, in Australia, that ratio is flipped, and they have about 70% virus negatives in Australia. Lots of fried, fair skin in Australia, and they don't need the virus. Most of them are caused by UV there. But in the US, it's about 80%. And so we took about half and half. Well, there's 3 studies here that picked apart these really interesting genetic mutations. It's really night and day between how the cancer arises with a virus versus just being fried by UV. Here's one of them, mutational landscape of Merkel cell polyomavirus negative and positive cancers with implications for immune therapy. The distinctive mutational spectra of polyomavirus negative Merkel cell carcinoma and UV-associated mutations underlie the etiology of Merkel virus negative Merkel cell carcinomas. So what did we find we had, again, split them roughly in half. We put about half that were virus negative, half that were virus positive, and did exome sequencing. So the exome is the 3% of our genome that is encoding for exons or for mRNA. And so that portion was sequenced. It's a little less expensive than sequencing the whole genome, and that's where most of the action is. And then the tumor mutations that would then potentially be visible and functional were mapped. And look at this dichotomy. All the stuff on the right has essentially no detectable single nucleotide variants. Those are mutations. And this on the left has a really large number. And what is the flavor of those mutations? If you break them out based on UV versus age or other types of mutations, look how blue this side is. It's all sunlight. It's all heavily mutated and from sunlight. Well, what's going on this side, which doesn't have sunlight and doesn't have mutations? Polyomavirus positivity in terms of antibodies in the blood. And fascinatingly, when you map the mutations that happen, and the dots are mutations in P53 or retinoblastoma, critical tumor suppressors, and when you got a box around them, you've got a second mutation, a loss of heterozygosity. So you've really double mutated these. Look at how many double mutated things there are on this side, and look at this silence on the virus positive side. So you basically have virus negatives that are really highly mutated in all of these UV-induced mutations, and you've got virus positives that are quiescent in terms of mutations. But so we have a simple summary. All of those papers made the same conclusion. You either need a lot of UV damage to make a Merkel cell carcinoma, or you need the polyomavirus. And happily for patients, both of those things are visible to the immune system. Viral proteins in a tumor, as are mutations caused by UV. And I'll be showing you here superimposed on TCGA, the Cancer Genome Atlas. Many of you have probably seen this slide of all of the types of solid cancers. And here are the least mutated ones, thyroid carcinoma and pheochromocytoma and such. And the most mutated ones, lung adeno, lung squamous, and skin cutaneous melanoma. The Merkels that are virus negative and have UV mutations are almost a log more mutated than the most other mutated type of cancer. And then the Merkels that are virus positive are less mutated than anything else. So this is kind of cute. We have these little spheres, a virus causing this type of Merkel and a sun causing this type. And this is 10 to the 8th meters big, and this is 10 to the minus 8th meters big. So kind of a cute comparison there. But the bottom line is there's potential antigen visibility to the immune system in either case, which is life-saving for our patients. So how visible is this virus now to B and T lymphocytes? So here's where some of the folks in our department become, again, really prominent. Kelly, Mark, who I see in the audience, and Denise and such. We found that humoral immunity is a very powerful biomarker. So Kelly, this is one of Kelly's 35 publications on Merkel cell carcinoma. She did an MD PhD in 3 years, and then most of her publications came while she was a medicine resident and such. She came into my office one day and said, "Paul, our assay has gone kaputski." That wasn't her word. And I'm looking at the same patient's blood, and we're getting totally different results. And I said something soothing and useless. And she went back to the lab and stared at her data for a little while, and she came back excited, and she said, "Oh, they're the same patient, but different days, different disease burdens on those days." And then she started digging through all these other patients and found that as the cancer gets cut out, those antibodies go down. Let me tell you just a tiny bit. Here's a picture. Here's an EM, electron microscope image of a Merkel polyomavirus virion, and all you can see is the capsid protein on the outside. This is the immunogen. This is what we in the room have antibodies to. 60% of us have antibodies to that today. In contrast, this little red thing that I just PowerPointed in is a reminder that there's a double-stranded genome inside that capsid, and very transiently, very sneakily, the virus will express that and then hide it as quickly as possible. Makes the cell divide and then goes away. And none of us have antibodies to that in the room today. But more than half, just over half of Merkel cell carcinoma patients at the time of diagnosis have these antibodies. And then the fascinating thing is that the capsid antibodies stay rock stable over time. So these are 10 patients who did not have their cancer recur, all different colors, and number of days from their diagnosis, and there's no difference. In contrast, the oncoprotein antibodies that were positive here fell really quickly and on average were gone by 8 months after their diagnosis. And you might ask, well, if they fall away like this, what happens if the cancer comes back? So this person had no evidence of disease at the time, at this point, when their oncoprotein antibody titer was up by a log, they had metastatic disease, and then that was treated, and then it went back down again. So, it was quite obvious that this might be clinically useful. Then here's a cute little photo we took in front of lab medicine there with Mark and his team. Developing a clinical antibody assay. We started this on May 28th, 2010, and the paper was just accepted this week. So it's a bit of a journey, but we did a prospective study going forward of antibodies to the Merkel cell polyomavirus oncoprotein. It's NIH-funded study. 465 patients enrolled prospectively with over 1,000 blood draws looked at, and 219 had their first draw. They met very stringent criteria to be very clean, had their first blood draw within 90 days of diagnosis, and of those we had 1.9 years of follow-up, and we drew blood typically every 3 months or so. So here are patients who had those titers falling. Here's 4 of them, for example. The day of diagnosis, and then 1 year, 2 years, falls, stays down. None of these patients recurred. And when we look in aggregate across 176 draws and 62 evaluable patients, 98% of those who had falling or negative titers had no evidence of disease around the time of that blood draw. In contrast, for those who had increasing amounts of antibodies, here no evidence of disease to metastatic, 88% either had disease at the time of the blood draw or soon after developed it. So very useful test. It was made available as of January 2014, run by UW Lab Medicine. Here's a little screenshot of the ORCA order. And in case you don't believe it, I couldn't believe it. It was really cool. And then here is it summarized on our website for patients who are using this literally around the world and their docs. And it's pretty inexpensive compared to a CT scan, no radiation, obviously, no contrast. And it helps both virus positive and virus negative patients. How is that? Well, at the baseline, I should say antibody positive and antibody negative patients. At the baseline, you just get this test, and if they're negative, they actually have a 42% higher risk of having the cancer come back. Either because they have a virus negative Merkel, which is a little bit more aggressive, or they're not making antibodies to their virus, which kind of is a bad indicator for how well their immune system is working. And then we know we need to track them closely with scans. And in contrast, if they're positive, we can follow them with serology, and we can really spare them a lot of scans, which is nice. So if antibodies are useful for tracking this cancer, what the heck are T cells good for? Surviving. If CD8 T cells are moderate or high within the tumor, if they migrated into the cancer, no one died of Merkel cell carcinoma. First in a cohort of 146 patients, and then a separate cohort of 150 patients. This is, and those were both more Kelly papers. And here's the action zone. How do CD8 T cells recognize their target? Here's a picture of the T cell receptor finding an antigen, a peptide, 10 amino acids roughly, from, for example, the Merkel polyomavirus, expressed in the groove. We call this the hot dog bun, and we call this the hot dog. The virus peptide is the hot dog, and the MHC molecule is the hot dog bun, and we have to explain this to patients. Can you imagine how painful that is? So we do the food analogy, and this is a tongue that can define 1 in a billion specificities of the type of hot dog and hot dog bun. And this is because some of our trials depend on what HLA type they have. So we have to explain this stuff to them. It turns out that 80% of Merkel cell carcinomas downregulate class 1 MHC, but happily we have figured out ways to re-upregulate that and get the hot dog buns back out there so that the tongues can see them. So how did we pick apart the tongues and the hot dog buns? Well, David Cole, the day after, probably the day of the release of this viral sequence in Science, he calls me up and he says, hey, you got to study this. And it's going to be straightforward. And he was right in many ways. So we take T cells from either the blood or the tumor-infiltrating lymphocytes. We made peptide libraries, mostly focusing on the only 20% of this virus that's needed for causing the cancer. Really convenient, small amount of real estate, immunologic real estate to be studying. That translates to only 95 peptides, even if you tile them overlapping like this. This is literally the 95 peptides that we need to cover those 389 amino acids that are responsible for the cancer. And we then just ask, well, with this peptide, do those T cells get excited and secrete interferon gamma or not? And this led to the first definition of the first 26 epitopes from the virus, and all these guys that turned red, you can see shotgunned across the real estate there, were pieces of the virus that our human immune systems can recognize. So that also generated some very powerful tools for studying Merkel cell polyomavirus T cell responses. This is an MHC peptide tetramer. So to the patients, we got a little glowing orb in the middle, a fluorophore, and we got a hot dog bun and a hot dog in 4 copies linked to this glowing orb. And that has the capacity to label up 1 in a billion specificity T cells and allow us to ask, where are they? What is their functional status? How many of them are there? And so here's one of the first questions we asked. With this particularly effective and easily seen epitope, in control subjects who were HLA-A24 positive, had that same hot dog bun, no one had any T cells that bothered to recognize this virus. In contrast, in Merkel patients, 2/3 did. So if you're with me here, why do virus-positive tumors grow if patients have pretty good numbers, in many cases, of T cells that can see them? They should be getting blasted by these little grenades from the CD8 T cell, the granzymes and such. They should be killing the virus-infected cells, and they're not. Well, are virus-specific T cells dysfunctional? Using our tetramer, we can ask that question quite straightforwardly. First, I'll remind you, though, that T cell surface receptor phenotypes now can be an indicator of what the activation or dysfunction status is of a given T cell. So when you activate a T cell initially, they express these little green guys, these proteins. If you're not an immunologist, don't read the stuff on the right. But they turn green. All right, I'm kidding. But when they get chronically stimulated, T cells aren't happy after 1 or 2 or 3 weeks or something like that, and they express these red guys. PD1 and TIM3 are the relevant ones from this slide. And of course, reality is more complicated, and many fit somewhere in between. But by the time you're expressing multiple markers of dysfunction, you probably are dysfunctional. And we asked how many of the T cells that recognize Merkel cell carcinoma are dysfunctional and express double positivity for PD1 and TIM3. And we used controls from the patients themselves to their CMV-specific T cells, cytomegalovirus or Epstein-Barr virus-specific T cells from the blood. And you can see baseline, not so many are dysfunctional with that, 5% or something like that. In contrast, whether the Merkel cell polyomavirus specific T cells were in the blood or they were in the tumor-infiltrating lymphocytes, much more dysfunction. And what does that mean? That means we should be able to target these, quote, exhaustion pathways with PD1 and PDL1. So this was the rationale, and here is a quick reminder of the brakes and accelerators that control the T cell response. Here is signal 1 and here is signal 2. Signal 1 is the key interaction that gains all the specificity between the T cell receptor and MHC peptide, our hot dog bun combination here. In signal 2 is a critical B7 CD28 signal of co-stimulation, which is disrupted by this dominant molecule that comes up, CTLA4, and pushes the CD28 out and blocks co-stimulation normally. This interaction can be interrupted with anti-CTLA4 antibodies, also known as ipilimumab or Yervoy, the first major immune therapeutic for melanoma about 6 years ago now. And this pathway, this exhaustion pathway, as soon as a T cell gets activated, it upregulates PD1. And it says, well, if somebody out there needs to turn me off, this is how you do it. And the PDL1 then binds to it and shuts off the T cell, and this can be blocked with either anti-PD1 or anti-PDL1. Big blockbuster drugs, Opdivo, nivolumab, Keytruda, pembrolizumab, and such target this area. So I'm just we've had a lot of clinical trials going on, very interesting in terms of biomarkers and outcomes. Shalender and Ode are both members of our medical oncology division and are close collaborators and have led multiple of these trials. I want to talk mostly about two here that have now just been reported and are really changing the landscape of management. So Mac Cheever is the head of the Cancer Immunotherapy Trials Network at the Hutch and in medical oncology at UW. And together with Mac, we designed an anti-PD-1 trial in Merkel cell carcinoma, and it was a bear to get a pharmaceutical company to give us the time of day. Rare cancer they've never heard about, no real market, blah, 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 blah. Chemotherapy will shrink it in 2/3 of cases, give me a break. It was not an easy go. So we teamed up with the NCI, with CITN, and we kind of leveraged Merck into saying, okay, well, they didn't have a choice. They already had given the drug to the CITN, and so if CITN decides they want to do something, they can do it. So we ended up doing an 8-center, single-arm, open-label, phase 2 trial of first systemic therapy. We made a strategic decision, no chemo before trying the immune system. We know chemo suppresses T-cell function and makes the cancer more aggressive. So this was another thing pharmaceutical company wouldn't like, because we're going head-to-head against chemo, which works 60% of the time. And that's not a good combination. And they also refused to do a randomized trial between chemo and immune therapy. So we tried to do that. Unfortunately, now it's not possible because these results are so good. But I will show you why it's probably okay later. But these are the machinations that go through your mind. You're trying to work with, how do you design this stuff? So we ended up using pembrolizumab at pretty standard dosing, and the objective response rate by RECIST criteria, just tumor shrinkage, was the endpoint. And so this came out in June, PD-1 blockade with pembrolizumab in advanced Merkel cell carcinoma. And again, really important collaborations across the government and eight universities and Merck. And here's the waterfall plot showing the extent of shrinkage in these cancers, color-coded by negative viral status in orange and positive viral status in purple. And you can see they're kind of random. They're all over the place. You can have patients who had big increases, more than 150% in their tumors, and I'm actually going to show you a picture of this guy in a few minutes. Or you can have great responses, complete responses, or deep partial responses, regardless of the viral status. There was a trend towards the virus positives maybe doing a little bit better. That's probably real, but we don't know yet. So here's maybe a more interesting graph. This is the spider plot or the spaghetti plot. Each patient is a line, and each time point is shown, each scan is shown by a dot. And again, we've got the same color scheme, and you can see people do well or poorly, although mostly well, with this therapy, regardless of their viral status. And really, it's a fast response and then a very stable one. Either you just progress like you're giving saline, or there's a really meaningful deep response quite quickly. And I'm going to point out here, not randomized, obviously, as I told you, but this is where chemotherapy poops out. 3 months. We'll come back to that, but you can see these responses are looking really good. Here is the swimmer plot. These are responding patients, blue for partial responses, green for complete responses. Some of them progressed. The red dot at the end is the disease coming back, but most of these responses were very durable. 86% were durable, and still, I can tell you now, almost 2 years out, they're mostly very durable. And then there's this fascinating thing that teaches us something about the immune system. Even 1 or 2 doses of this drug can profoundly change the equilibrium between the cancer and the immune system. This patient got 1 dose, developed a side effect, had to come off of it, gets a complete response, and now this line is out to here. Actually, it's way out here. He's almost 2 years out. There's no cancer coming back. 1 dose. This one had 2 doses in the same deal. He's in a deep, deep partial response in liver metastases, which is a semi-immune privileged location. So we're really changing, in some cases, the long-term equilibrium between the cancer and the immune system. So at least there's some hope that patients can get off of this. Probably a question is like, well, how long are you going to have to treat? We don't know the answer to that. When patients have side effects, we stop, and a lot of times they do very well. Here is the progression-free survival curve. Actually, it's out longer now, and actually it's above 50% at this point, which is really awesome. But I want to just compare the chemotherapy historical data. That's what it is at 3 months comparatively, 6 months, 9 months, and 12 months. So there are big problems with not randomizing and putting considering patient populations together because of known confounding factors, and per the FDA, the unknown confounding factors are even a bigger problem. However, once you start getting to a 3-fold, 5-fold, 10-fold difference, I think we can feel confident this is real and different. Here's one case, a woman who presented with her Merkel on the knee, and 10 months later developed bulky tumors in her pelvis, meaning she had to go, sort of presented by having to go to the bathroom every hour, no sort of bladder capacity left, and a bunch of subcutaneous metastases on her leg. One dose, comes back the first visit, she said, this thing was almost gone in 1 week. I did a biopsy after 1 dose, and these are the results at that site. You can see clusters of blue. That could be cancer, that could be immune cells. They stain with CD45, immune cells, and there's not a single cancer cell left marked by CK20 by 1 dose 3 weeks after. So a pathologic complete response after a single dose of pembrolizumab. And what happened in her bladder? Here's her baseline. Her bladder's squished into a little horseshoe, and she's got all this bulky adenopathy all over here. And by the time of the first scan, she's not going to the bathroom every hour and feeling really well, and she's out now almost 2 years as well. So in summary, anti-PD-1 in the first line, almost a 60% response rate with advanced Merkel cell carcinoma. We got responses in both virus positive and negative cases, and of those responses, they're typically really durable. We're trying to figure out, the simple answer is we can't predict. We've tried 20 different things. We can't predict who's going to respond and who's not. So you could say that's a glass half full, half empty thing. We got a pretty good therapy, but we don't know who's going to respond and who's not. This is now being expanded, hopefully for FDA approval, to include an additional 24 patients in a confirmatory study. This is a separate study done by EMD Serono and Pfizer that several of us were involved in, Shalender and myself, across, this was like 60 centers around the world. They put real money into this, and they chose to do it in the second line where things were hopeless, second or beyond, had to have failed chemo, you have no hope. So these folks all did really well here on this side, but I want you to notice the right side is redder than the left, which is blue. And what does that mean? 1 previous line of systemic therapy is red, 2, 3, 4 lines of failed chemotherapy is blue. And my gross oversimplification is that this is your likelihood of responding. If you have had 0 rounds of chemo, if you've not failed chemo, you've got about a 60% response rate. If you failed once, 40. If you failed twice or more, about 20. Not an advertisement for what the immune system, the effects of the immune system, of chemotherapy on the immune system. So here's an editorial that came out at that same time talking about both of those studies. Checkpoint inhibitors, a new standard of care for advanced Merkel cell carcinoma. Taken together, these reports strongly suggest that checkpoint blockade is the best option to treat patients with advanced Merkel cell carcinoma. And happily, just a couple weeks ago, the NCCN guidelines came out for 2017, and they list pembrolizumab. So that's a very important thing. In some ways, people say more important than the FDA because it's really just about the money. And if the NCCN guidelines say pembrolizumab is indicated, insurance companies typically need to pay. Also, the companies are being very generous with giving out these agents free of cost if insurance denies it. So half of our patients do well on anti-PD-1. What do we do with the failures? There's also some patients we can't treat because they have autoimmune disease and such, and these drugs tend to make that worse. Well, for those who fail, doing what has worked in melanoma has worked sometimes for us. That is adding CTLA-4 blockade, ipilimumab. Other cases, we've used adoptive T-cell therapy led by Aude Chapuis. It's very encouraging, exciting results. We combine pulling out their virus-specific T-cells, growing them up, and combining them with these broader, immune-stimulating drugs. So far, really good results from that. And if not, that though isn't possible if you don't have T cells in the first place that are either specific in their type or sufficient in number. And so what we're working with Phil Greenberg and Ode to do then is we've found fantastic, super high affinity T cell receptors that recognize the most common epitopes. And those people who have those really good, high, sticky T cell receptors do much better. We can now stick those in en masse through a viral transduction into the patient's cells and reprogram them to be highly specific for this virus. And that's what we're working on doing very soon. The last thing I want to show you is how we've rescued patient with natural killer cells. This is a trial going on with Schilender. This is that patient number 1 who failed spectacularly pembrolizumab. He had lesions on his scalp. This is what 12 weeks on pembrolizumab did to this guy. Nothing good. He got some radiation that controlled some of them, but he had recurrences, and then he got, crazily enough, there's one natural killer cell line that they just grow up and irradiate and stick into anybody. And it doesn't cross-react, really. It looks for things that shouldn't be there, like tumors and such, and then the cells die because they got radiated. Speaks for itself. This is somebody who failed pembrolizumab. So this is innate immune system, and in the future, we will not just be using the adaptive immune system, but we will combine innate immunity in intelligent ways to rescue the people for whom adaptive immunity, T cells directly, aren't cutting the mustard. So the vision for future management of this cancer, we want to minimize recurrence through early, best management, stimulate the immune system, perhaps in an adjuvant setting. We're getting ready to do a 100-patient multi-center trial. There are patients that have an 80% risk that the cancer is going to come back, and we're doing nothing for them now besides observing them. So those patients are great candidates for an adjuvant trial, which is getting started. And then if they recur, find them early, reduce the tumor, reverse the immune evasion, and figure out good and better ways to stimulate the immune system. So I'd like to thank funding sources and wonderful collaborators across the Hutch, UW, the Benaroya Research Institute, and the core facilities at the Hutch, and lab medicine here. Wonderful clinical team. This absolutely is a multidisciplinary cancer. You need radiation, you need surgery, you need medical oncology, and as a dermatologist, we insert ourselves and try and help manage the patients. And then a wonderful lab team. I talked about many of these people, and Kelly is just a few months away from starting back in the lab, which she's very excited about after finishing her Nghiem fellowship.